Okay, I think we can get started. All right, so this morning I mentioned a couple of times yesterday, and you heard all week how important we think it is to have this prior knowledge in terms of an assay library. We wouldn't say that much detail about how to make these, and this is the sort of purpose of this lecture. So I just remind you, I think you should be drilled into your brain already about why we care about assay libraries, and then we'll walk through this procedure, that the way that we do it here at least. I'll say a few words about some publicly available libraries, and I already mentioned this sort of 10,000 protein combined human assay library that we published not too long ago. And then make a few comments about false discovery rates when you're applying these libraries to large, to SWATH. So when you're applying these large libraries to SWATH data sets, and some special considerations that you might want to think about. And then maybe a few comments about library sort of portability, which this topic has already come up a few times. So here's our complex. So I mostly give this from the perspective of SWATH analysis, but the procedure and the concepts are very similar for SRM, but I'm going to go from the SWATH side. So here's again is our complex SWATH-MS2 spectrum from 600 to 625 with all these complex fragmentine spectrum arising from multiple different precursors that we cannot interpret in a classical way. And I said, okay, we know there's a peptide in here, we know these are the major fragmentines and they're colored in here, but you would not be able to find these if you didn't have some way to deconvolute the spectra or to cut through essentially the interferences. And the way I said we did this is by having a library spectrum, so a pure spectrum which belongs to this peptide. And when we get this, we can do this targeted data extraction and get to selective and specific data on this peptide from this complicated SWATH spectrum. And here's the table, right? This is what we want to have at the end of this exercise. We don't need the SRM stuff. It's just the protein accession, the peptide sequence, the precursor and fragment masses, other details about the precursor and fragmentines, and also the relative intensities are very important, and the normalized retention time. So what kind of motivated us to think about this was, you know, when we started presenting this SWATH principle at meetings 2 to 3 years ago, maybe 4 years ago, we would go out, or Rudy would go out, and people would say, you know, we would try to make the point that if you have some SWATH data and if you have an assay library and you kind of put these together and do a targeted analysis, then you get nice, complete, sensitive, robust quantitative data, which is what we've been trying to convince you about. And then everyone would always say, oh, okay, but where do you get the assay libraries from? There's a lot of kind of confusion about this topic. And some people who had done some SRM or done some more classical targeted proteomics had some idea about this, but the more shotgun DDA-oriented people found this a bit confusing. So this kind of motivated us to write this down. I should say this motivated Rudy to motivate us to write this down in a protocol. And we did this recently. So what I'm going to tell you about is basically described in much more detail in this protocol paper. And the goal here, I'd say, is twofold, right? So we wanted to write down a protocol, which is some ideas about what we think might be the best practice to do this, but also as a means to kind of document how, if we're going to distribute libraries that the rest of the community can use, then we've kind of documented in detail how we've made these, and we don't have to write it down every time that we do it. So the basic workflow is that you take some of the representative samples of your sample of interest, and you do kind of a classical DDA experiment, shotgun experiment. Then you need to convert this data, and well, mainly this step is peak picking. We usually convert it into these open source MZXML, MZML type formats. But an important step is peak picking here so that you can give it to the search engine. Then we do a database search. We apply some false discovery rate control on the search results. And then we take the spectra and we combine these into a spectral library. So this is an important step. What you'll find is that when you do these types of experiments, you have redundant peptide spectrum matches. So by that I mean you have multiple instances of spectra that identify the same peptide, and you have to, there was a bit of discussion about this already, you have to decide if I have 20 spectra for one peptide, what am I going to do with these? Because I only need one representation of this peptide in the library. And then this spectral library gets filtered, into an assay library. So this, I'll say what this means, but it essentially means selecting the appropriate set of fragments that we wanted to monitor in a targeted analysis. And this kind of simplified workflow is described in this protocol in a rather more complex format. And I just want to take sort of, a few minutes to walk down through this. There's a lot of details and steps here, and I won't go into describe at every, very low level of detail. I just want to say what each step is and a couple of things that you might want to watch out for. So this is the same again, right? You have some kind of sample preparation, data acquisition. We have some converting and centroiding going on here. You have your search engine and some post-processing of the search engine we use these TPP tools, Peptide Prophet and iProfit. The search engines we use are typically the sort of open source ones, XTandem or Comet, but it can be whatever you want. This combining the spectra into a spectral library is done using a tool, also part of the so TPP is Trans-Proteomic Pipeline. This is a set of tools from the Institute of Systems Biology in Seattle that are very popular for shotgun data analysis. And Spectrast, Peptide Prophet, iProfit are all part of this tool set. Spectrast is the one that we use to reduce the redundancy in the spectral library so that there's just one instance of a spectrum per peptide, which is what we need. And then finally, we have essentially want to go from this set of spectral libraries into this table format that I showed a few minutes ago, and this is just more or less a matter of filtering, selecting a set of rules similar to what you did in Skyline tutorial, saying we want to pick, 6 Y ions, the most intense ones, or something like this. And at the end, you should have a table, either a table in a kind of CSV format, or there's another file format called TRAML, which is an XML-based format that is a sort of a standardized format for representing these assays. And these Skyline, Spectra, and other PeakView will all import this table format. For OpenSWAT, you need this TRAML format, and you can also optionally add decoys at this stage for OpenSWAT analysis, whereas in these other tools, the decoys are kind of generated on the fly, or internally to the software. So I want to point out that this is just the workflow that we use and that we've described. There are other ways to do this, right? You can use your favorite search engine here. Skyline will handle basically this whole part of it, and will accept, we'll be able to import data from many different search engines. And so the way what's written in the tutorials is not exactly what I'm describing here, but as yesterday when we talked about OpenSWAT and Skyline, in a conceptual sense, they're doing more or less the same thing. And maybe I'll point out one or two places where they do something different, but you can do it either way. And the idea of having this more detailed protocol is that you have control at each stage, and you can kind of understand what's happening at each stage. So first of all, the idea is to get, you need to select a set of samples, right? So this can be some representative set of samples from your experiment. Some samples from different conditions. If you have a perturbation experiment, you could select samples from across the perturbation, for example. You can optionally do some fractionation of these samples to go a little bit deeper in the library. In the case that we know that the DDA analysis is often not quite as sensitive as the SWAT analysis, and so we found in some cases it might be beneficial to do a fractionation step here for the purpose of the library generation, so that you have whatever's you have a more comprehensive library, and you have a better chance of extracting all the signals that are detectable in the SWAT data. Another common approach is to synthesize peptides. So I think somebody mentioned already this week that you can synthesize crudely synthesized peptides, so not pure and not where you don't know the concentration, for about $10 or something like this per peptide in a 96 well plate format, and this is also a common approach if you want to, in particular, if you want to have spectral libraries for proteins and peptides that might not be amenable to detection using the more classical DDA approaches, or may not be represented in the samples you have available when you're building the library. Important to add some kind of retention time peptides. This has been discussed a lot already. This is particularly important for the scoring in this targeted data analysis, but also for sort of portability if you want to move these libraries around between labs and so on. So a couple of things to maybe point out for the data acquisition. The settings that you put on the instrument to acquire these MS2 spectra may not be identical to the settings you would use in a standard DDA analysis, because the goals are slightly different, right? So in a standard DDA analysis, you want to just try and identify as many things as you possibly can. But in this case, what you want to do is have a library spectrum which is basically representative of what you're going to measure in SWATH. And one way to achieve, so you want the relative intensities of the fragment ions in your library spectrum to match the relative intensities in the SWATH analysis. And one way to get higher signal to noise in the MS2 and in the DDA analysis is to increase the accumulation time a bit. So for example, on the 5600, I think AB recommends you use 50 milliseconds or something like this to maximize the number of identifications. Typically, we push this a bit higher, and this gives you a higher signal-to-noise spectrum in the MS2, and it's you have a better chance of your relative intensities being from the DDA spectrum being in good agreement with the relative intensities from the SWATH experiment. It also can be beneficial to collect more than one MS2 spectrum per peptide, because this idea of kind of averaging spectra or making consensus spectra helps you with this signal-to-noise problem as well, and also so the representation of the relative fragment ion intensities when you sum over more spectra for the same peptide is going to improve this too. And I think this came up already yesterday, but it's nice to, so in SWATH we said, Jared said yesterday, or in DIA, you can't customize the collision energy for every peptide, because you know what the mass of the peptides is approximately, right? You know which isolation window it's in, so it's somewhere from 600 to 625 in that last spectrum I showed, but you don't know what the charge state is, so you cannot exactly customize the collision energy for every peptide, so we kind of have to take a guess about this. And what we said yesterday is that we usually calculate the collision energy for a +2 fragment ion around the mass of this, the middle of the mass range of the precursor isolation range. So if it's 600 to 625, then you choose 610 or something, calculate the collision energy for a +2 ion, and this is basically what you apply. The thing is that when you do the DDA analysis, now you have usually an equation that determines the collision energy, right? So it detects a precursor, it says, oh, this is a +2, it knows what the mass is, and it uses some equation to compute what would be the optimal, the machine thinks the optimal collision energy would be. And now we have a disagreement between the collision energy that we have for the DDA and for the SWATH. So what we rather do is take the SWATH collision energies and apply them in the DDA settings. So in our DDA analysis, when we're generating libraries, we pretend everything is basically a +2 ion, and we just compute the collision energies for this. So we've done a little bit of optimization, and we have an idea that these things are going in the right direction. Also, a colleague of ours in Seattle, Sam Bader, did more work on this optimization of IDA acquisition settings to build SWATH assays, and he's written a manuscript about this. I think it's submitted, but I think these are basically the ideas that are encapsulated in what his paper says. So when you're converting the data from the raw format that the instrument spits out, usually there's a step, or else sometimes it's done in acquisition time, depending on which instrument you have. But on the 5600, which we are mostly working on, the peak picking and centroiding is done post-acquisition, right? You collect profile mode data at acquisition time, and there are many different ways to pick the peak, right? So we need to get a list of masses to give to the search engine, and different algorithms do it different ways, and some of them do more or less processing of the spectra and go a little bit further away from the raw spectrum. And this can have a benefit in terms of the performance of the search engine, but it may not be what you want if you want to keep the spectrum the same and still in good agreement with the swath, right? So here's an example of this. 3 different peak picking algorithms, and these are exactly the same scan, right? So this is one MS spectrum that we're applying 3 different peak picking algorithms to. This is the one that comes with this ABCIX converter in so-called protein pilot mode. So protein pilot is the search engine from ABCIX, and if you want it, their claim is if you want to maximize the search results, you should put it through this peak picker, and it's going to do something different to the spectrum than if you chose just the regular. There's also another mode in this ABCIX converter where you don't have to do this additional processing. This is MS Convert, which is a program that Brendan mentioned earlier, which also has these peak picking algorithms, and it has available to it also the vendor algorithms for doing the peak picking. So this is run in this open source MS Convert software, but it's actually using the ABCIX libraries essentially to do the peak picking. And this is a tool that was written by a developer in our lab called QTOF Peak Picker. One main difference here is that it's taking the area, these intensities of these peaks is computed by taking the area under the peak essentially, and not the peak height. And we think that this matches better to the swath data. So you can see that even though these 3 spectra come from exactly the same original raw spectrum, they look quite different in terms of their relative intensities. And this is kind of an extreme case, but if you now look at how the relative intensities look in the swath analysis, what we see is better agreement between this kind of in-house developed peak picker. Well, actually it was specifically developed to agree with the swath data, so it should, but earlier we were using these, and there are instances when they don't work very well.
Sorry.
Yeah.
So when you say the area under the curve, that means under the profile points, not
exactly.
because you don't have multiple MS2 spectra.
Right, exactly.
Okay,
so I mean, the peak height as compared to the area under the profile, exactly that.
Yeah. Okay.
Yeah. So and if you think about it, when you're doing a swath analysis, you're doing extracted ion chromatogram extraction. That's also what you're extracting when you go to do it in swath, right? You draw some borders and you take the area under the profile. So it's kind of, it's a like for like comparison then, and this is one reason why we think it maybe is working better. Yeah, so we get better agreement with the swath data, and this is essentially what we're looking for. And this is now a bit more larger scale comparison of these peak pickers. Yeah.
Sorry, just one quick question to go back to the previous one. Mm-hmm. So the area that is picked, is it for the whole isotope cluster or for the monoisotopic peak?
It's for the monoisotopic, because this is, again, also what we extract in when we do the swath analysis. We just take the monoisotopic. Yeah.
Stop.
Oops.
If you go back one slide.
Yeah.
Actually, so this is for 5600 data, I guess, right? So
yep.
for TOF data.
Yep.
Have any recommendation for QExactives, which algorithm to pick?
I don't. So I think this would be a good question for Jarrett when he comes back, or for Lucas Reiter, who will also, these two guys have done a lot of swath analysis, swath type analysis on QExactives, so maybe we can hold that question for them. Okay. And so something we looked at also is, there was a master's student in our group. We were interested in this question about comparing similarity of spectra. So if you take 2 spectra and you sort of overlay them, how similar are the intensities, the relative intensities? And a master's student in our group wrote a tool basically that kind of does these comparisons. And if you're interested in doing this kind of thing, it's nice, kind of like user-friendly Python GUI tool. So you can take spectral libraries and take one, input the other, and do a comparison. And so we did some comparisons of these various peak picking algorithms, and also in swath. And this is what I call the intrinsic variability. So if you take one sample and you inject it 3 times, and then you do the centroiding with this algorithm, and then you compare these 3 samples, how well the DDA spectra internally agree with each other, essentially. So you have, right, 3 different files all picked with this one peak picker. And this score essentially on this scale is a normalized contrast angle, it's called. It's essentially like a dot product, right? The score says how well these 2 spectra agree with each other. And so if you take this protein pilot ABCIX algorithm compared to MS convert, compared to the QTOF peak picker, we get better intrinsic, so over multiple spectra of the same peptide that we have across different files, there's better agreement in the relative intensities of the ions of this when you use this QTOF peak picker. And actually you can also do this comparison for SWATH as well. So we take multiple SWATH files, we look at the relative intensities across different files and check how well they agree with each other. And in fact, SWATH is the most stable one. Does anybody know why that might be? Why the SWATH essentially estimation of these relative intensities might be more stable than the DDA estimation? So one reason might be that in this SWATH comparison, we took the area under the chromatographic peaks, right? So we took each of the, say, top 6 most abundant fragment ions, compute the area under the chromatographic peak, and then this is what we use for the relative ion abundance estimates. And this is also what we do in, well, I think this is, so it's not done based on a single spectrum, but it's based on multiple spectra over the chromatographic time averaged together. So you can imagine that this might be more stable than the DDA method, which takes only a single spectrum, right? You only have one instance of this spectrum. Another comparison we can do is, in this plot, by the way, I should say this little S number here is important. So this is a dissimilarity score. So the higher this number is essentially, the lower this number is, the more similar the spectra are. I don't know if you can read that, but the second question you can ask is how portable are the assays generated in DDA and processed with any of these peak pickers? How well do they agree with SWATH, right? So this is how well do they agree internally, and this is how well do they agree when you go from DDA to SWATH. And we can see here that the differences are a little bit smaller, but essentially the QTOF peak picker that was developed in-house performs a little bit better. And this is not entirely unexpected, as I mentioned, because this was kind of optimized for this purpose, whereas these 2 were probably more optimized for getting best DDA results, maximizing identifications. Okay, then search engine, I think I don't need to go into any detail, but there's tons of search engines out. You can use whichever you want. We use then this transproteomic pipeline, this peptide profit and iProfit tools as post-processing of the search engines, and in particular iProfit allows us to combine the results of multiple search engines, so this gives you some additional confidence. I've already mentioned Mayu a couple of times, but it's important that we control the false discovery rate also at the assay library generation level. And I try to illustrate this with this point, right? So the question is, what happens if you make an error in the DDA analysis in the library generation? Will it get propagated into the SWATH analysis? So let's imagine that this is a shotgun spectrum, an MS2 spectrum, which is kind of like, for some reason, not that high quality, and the search engine assigns the wrong peptide sequence, right? So the search engine makes a mistake. The question is, how will this get propagated when you go to use this in a SWATH analysis? And it kind of depends what kind of mistake that the search engine makes. So if these peaks all come from a single precursor and you just assign the wrong sequence to them in the search engine, and you say, now I'm going to use that assay to extract data from my SWATH data, this same peptide is going to be in the SWATH data, right? Even though you've called it as the wrong sequence. And the open SWATH or the skyline analysis is going to say, oh, these all colute really beautifully, and there's very good agreement between the DDA spectrum and the SWATH spectrum, so I'm going to say that's the right thing, but you've already called it as the wrong sequence here, so this is a problem. Another possibility is that the wrong peptide sequence is assigned in the DDA spectrum because you have now a chimeric spectrum, right? So even in DDA analysis, it's very common that you co-isolate 2 peptides, and now we have a mixture spectrum here. Well, so you still assign the wrong peptide sequence, but for a different reason. So then what happens when you go to the SWATH data? You try to extract this as one peptide, and what you find is that you get 2 distinct peak groups in most cases. So usually they won't exactly perfectly colute, right? And then they will get a bad score from OpenSWATH or from Skyline or whatever. So the SWATH analysis is somehow resolving the error that the shotgun analysis has made. The problem is that we don't really know what's the prevalence of these two different kinds of errors, actually, to be quite honest. So it's rather safer to assume that this is happening more often. And the point of this slide, what it goes to is how should we quality control the DDA, whether this should be kind of strict or less strict. If you think this error happens a lot of the time, then you could potentially be less strict in the way that you filter the shotgun data. But my suggestion is that because we don't really know this, you have to assume that this is happening too, right? So we use a tool called Mayu to estimate the error rates associated with the data analysis, and you don't have to use Mayu. I mean, there are many tools, kind of also classical target decoy approaches, but essentially what I'm trying to point out here is that, and this is known for many years, is that the error rates at the peptide spectrum match, at the peptide level, and at the protein level are not the same, right? So this is, iProfit score is essentially, you can think of it as a search engine score, and where 1 is very good hit in the search engine, and this is on the y-axis is the false discovery rate. So as you relax this scoring threshold of the search engine, the PSM level FDR goes up at a moderate rate, the peptide level FDR goes up at a higher rate, but the protein FDR goes up at a much higher rate, right? So if you say, oh, I'm going to filter my data at 1% FDR on the peptide spectrum match level, this could lead you to have, you know, a 10% false discovery rate at the protein level. And the reason for this is, this was kind of mentioned already by Brendan, I think earlier, is that in this kind of classical target decoy approach for shotgun data analysis, so each one of these little circles is a peptide spectrum match, right? And each one of these hexagons is a protein. And the green ones are the good ones, the red ones are the false ones. And what happens is that the good hits tend to build up on one protein, on small number of proteins, whereas the bad hits tend to be distributed randomly across all of the proteins. So this has the effect that as you add more false matches at the PSM level, as you relax this threshold, you get this kind of differential kind of creeping of the error rate at different levels. This is an important concept actually also for SWATH analysis, but what I'm trying to point out is that it's important to evaluate these results at all of these different levels, and don't just say, oh, I'm going to use 1% PSM FDR, because it could lead to very significant errors at the protein level. So in this graph, for example, if we took 1% protein FDR, to achieve this 1% protein FDR, we have to have a peptide FDR of 0.2% and a PSM FDR of peptide spectrum match FDR of 0.08%. So we have to be very strict here in order to achieve a reasonable kind of protein false discovery rate. And this is all published in this paper from Lucas Reiter a few years back. Brendan also referred to this, and this is kind of one of the papers that was important in this going in the direction of controlling the DDA error rates properly that Rudy mentioned on Monday. Okay, so next step, we've done the acquisition, the sample prep, the peak picking, we've done a database search analysis, and now we need to take this database search result and we need to somehow combine the spectra together. So as I mentioned, so for a given peptide ion, you probably have multiple redundant, it's very often the case that you have multiple redundant spectra, so up to 10, 15, 20, or 100 spectra that all identify the same peptide, and we need some way to kind of put these together. This is one place where Skyline and our protocol behave in a slightly different way. So Skyline we use this tool called Spectra from the TPP. Skyline uses a tool, also a spectral library tool called Bibliospec from the McCoss lab. And one difference in the way that they operate is that, as far as I'm aware, in Skyline and Bibliospec, they just take the best spectrum. They say, I'm going to just take the best one, and this is the best representation of the spectrum that I have. In Spectras, you have the option to do a consensus spectrum. So it means you take all 20 spectra that you have for one peptide, and you sort of make some kind of averaging of them. But it's not like a simple averaging. There's also some quality control. So if there's one outlier, or if there's a number of outliers, these guys will get kicked out. And what you also have is some kind of peak voting to decide whether the peak belongs in the consensus or not. And conceptually, this is an attractive idea, right? So if you have fragmentines so this goes to this matrix question yesterday, maybe you have an interference in a certain matrix, but a different interference in another matrix, and by averaging over these, the hope is that you will eventually get to a spectrum which is just representative of the peptide and the best fragmentation, relative fragment ion intensities that you can achieve for that peptide. Also, I point out that this IRT transformation is done at this Spectra step, so Spectrust will go in and will look for the IRT peptides, which will be hopefully also identified in shotgun mode, and we'll take the retention times of these peptides, we'll do the linear regression, and it will return IRT values for all these peptides, and we store the retention times in this normalized retention time space that you've heard about a few times. So this is kind of what I just mentioned about the idea of either taking the best replicate spectrum, like what I think this is what Skyline does, or the consensus spectrum, which is more like what we have in our workflow. We've done a few comparisons of this using this tool. Actually, in the end, in the comparisons we've done, the differences are not huge. So if you can see here that the difference between the best replicate and the consensus approach in this intrinsic variability, so how internally consistent are the spectra, it's kind of pretty similar. You can see there's a little bit difference in the consensus spectrum. Again, SWATH is still the most internally consistent. And then we can also do the comparison from DDA to SWATH, whether the best replicate or the consensus agrees. And again, it's not a huge difference in this particular data set. This data set is very homogeneous. It's just a few yeast files. If you went to bigger data sets, I think there would be a bigger difference. And I mentioned this paper from Sam Bader in Seattle. He's looked at this a little bit in more detail, and what he says is that the number of spectra you add to the consensus also makes a difference. So if it's 1 or 2 or 3 or 4 or 5, the quality gets better up until around 15 spectra, and then it levels out. So you can see in this, it's slightly better in this dissimilarity score, but not a whole lot. But in other data sets, it might be more significant. So just to be aware that these two options are available. And if you do the default workflow in Skyline, I think you're going to get this one. So I mentioned already at this step, this linear regression is being done by Spectrust automatically if you select this. And so we're going from the real retention time space through this linear correlation with these IRT peptides, and we're storing this in this IRT space, and this really goes to assay portability, right? One sort of slightly aside on this IRT thing. Many people often ask, do I really need to spike in these peptides, right? Can't we use some endogenous peptides to do this recalibration? And the answer is basically yes, as long as you're sure that these endogenous peptides are going to be in all the samples. They need to be everywhere, right? And we had a visiting scientist came to our group, visiting postdoc from Jenny Van Eyck's group. Her name is Sarah Parker, and she came with a whole bunch of SWATH data that she collected in her own lab, but there was no IRT peptides spiked in. And we spike, as was mentioned, we spiked these IRT peptides into all of our samples, and we kind of were like, oh, our data analysis pipeline doesn't know what to do with this. So she kind of came up with the idea of trying to make some kind of standardized workflow to use endogenous peptides to do this recalibration. She called it CIRT, so custom IRT. And what she did was she looked in lots of protein databases of several different species. She looked for very highly conserved proteins that were very commonly observed in like all mass spectrometry experiments that she could find. So these tend to be like filamentous proteins like actin and stuff, and also trypsin and keratins, common contaminants that you would expect. And she basically tried to find sets of peptides that she could find in every file. And she came up with some lists of these, and these kind of work pretty effectively for doing this IRT recalibration. This is almost out, should come out in the next couple of months in MCP, I think. But so I'll point out that this is not the primary kind of workflow that we use. We still always spike in these IRT peptides because it's very convenient. And you can also use it for kind of quality control and so on, but there is another option in terms of using endogenous peptides is maybe just a little bit more work. Okay, so now we're at the last step, and this is essentially to go from these consensus spectral libraries to this table format, and we need to do some filtering here. So we have kind of set of rules that we would suggest here. We usually suggest to do about 5 to 6 transitions per precursor. Select these charge states in this range. Y and B ions is fine, especially in a label-free experiment. If it's a heavy experiment, then you need to maybe think about it a bit. Neutral losses are also acceptable. We usually exclude fragment ions that fall in the precursor isolation window, right? So in the spectrum I showed before, this was 600 to 625. And what you tend to get, if you choose a fragment ion that falls in that window, it tends to be very noisy because there's a lot of unfragmented precursor alpha.
Sorry, Ben, just a question to the SpecTrust.
Yep.
So how different are the spectral libraries generated by Skyline and SpecTrust? Are they really different? Would you...
Not so. So you can sort of get an idea from this slide, right? It doesn't. We're using now not Skyline, but we're using the best replicate option in Spectra. So this is the best replicate option in SpecTrust, which is similar to what Skyline does versus the consensus option in SpecTrust. So this is more or less the comparison you're asking. And so for in this data set, they don't look that different at all. But this is a very confined data set with only like 3 yeast runs that we used for the purpose of this library protocol paper so that people would have a small data set that they could do the tutorial with. We haven't evaluated this on a very large, my feeling is that it might be different when you go to a very large library that comes from very heterogeneous, different types of samples and stuff. I think then it would make a difference, but I have to say that we haven't really systematically evaluated this. Our expectation is that consensus should do a better job, but we haven't really done this systematically. And I mentioned this paper from Sam Bader in Seattle, which he looked into it a little bit more detail, and he found that there was an increase in the agreement between the relative fragment ions in the spectrum to SWATH as you add more spectra to the consensus. So that's kind of half an answer, but it hasn't been evaluated systematically.
Okay, and second question. So which retention time is picked? Do we average retention times as well for the spectral libraries? Is this just a PEC spectrum? Is it the same for the score?
Yeah, so yes, we do. But we do it after the transformation to IRT space, right? This is important. So in order to do this, they need to be on the same scale. And I think we're taking the median, and I think there's also even an outlier removal. And this is fairly stable. And this is implemented in, inside SpecTrust. Inside SpecTrust, yes. So as of SpecTrust version 5. We used to do this with some Python scripts or something, but as of SpecTrust version 5.0, this IRT linear regression is directly implemented in SpecTrust, and my understanding is it's median and outlier removal. I'm pretty sure. Yeah. Okay. Right. So I mean, these steps essentially correspond to these peptide and transition setting tabs that you have in Skyline. You're just deciding some set of heuristic rules that narrows down the library spectrum into a set of into an assay, essentially. So a set of fragment ions that you're going to use to extract. So this is basically what we're doing here. And there's more details about what you may and may not want to do written down in the paper. I just kind of picked out the most important ones here. For OpenSWATH, we would also convert to this XML TRAML format and add the decoys at this stage. For these other softwares, a table format, like a CSV format, it will import these. So as to your question about SpecTrust, I mean, Skyline will import libraries that you've generated in SpecTrust. That totally works. You don't have to, so you can jump in at whatever stage in the workflow that you want. Okay, so this is more or less what I said. This is the final output, this table, either in TSV for these softwares or TRAML for OpenSWATH. Okay, so maybe a short summary on this kind of spectral library, assay library building workflow. So this is the kind of, well, I was going to say simplified. It's not that simple, but it's simple than the other more simple than the other diagram. It's just, it's really a fairly classical DDA analysis with some post-processing stuff on the end. And one thing to watch out for is this relative fragment ion intensities from DDA. We should try to make these agree with SWATH, so watch out for processing by peak pickers and file converters, that these are behaving in a way that consistent with what you want. Collision energies, we try to match these from DDA to SWATH. try to get, you know, higher signal to noise, better quality spectra. These will agree better with the SWATH data. We do this by going sometimes for longer MS2 scan times or combining spectra in this consensus approach. Control the false discovery rate of the DDA data, right? So we want to avoid propagating errors into SWATH. It's good to transform everything into normalized retention time space. So this is for scoring, but also if you want to make it portable to different chromatographic setups.
So in practice, which FDR are you using and at which level?
So we usually say 1% protein FDR, and this return, so where can I go back?
Yeah, the Mayu figure.
Yeah, but I just wanted to actually say what the numbers are, right? So we say, I mean, it's important because the number that you actually need is the threshold for the search engine score or the iProfit or whatever. So we say, okay, Mayu, you know, Mayu spits out a table which tells you the equivalency of these different false discovery rates, and we say we're going to make a cut at 1% protein FDR. That gives us 0.2% on the peptide and 0.08 on the PSM, and in order to achieve these FDRs, we need to throw out everything that has an iProfit score less than this, right?
So why do you care about protein level? I mean, you quantify peptides.
We quantify peptides, but usually in most experiments, the end conclusion is made at the protein level, right? So there's almost always, except for some PTM studies or something like this, you almost always roll this up to a protein inference, so it will go back up to that level eventually after the SWATH analysis.
And regarding the right part, what about taking only peptides where you have two spectra? Because you say your PSM and the decoy, they propagate uniformly on the decoy space. So and they stack up on the target. So you could go for only if I have two spectra for one peptide. Does it help?
In most cases, not. So this is. I would suggest you take a look into this paper, the original Mayu paper. This was evaluated at this stage, especially for shotgun, and the conclusion was that by taking two peptides, so you certainly reduce the error rate by taking two peptides, but it seems to be a disproportion. You lose a lot of information as well. It seems to be disproportional. And actually, there are options in the Mayu output table. It already computes this automatically. What would be the false discovery rate if you did take this more than two peptides? So this is already. You don't have to ask for it. It's already there. You can take that information from Mayu and filter your data that way if you want. If you know it.
And then I would like to make just maybe one more comment just to make sure that you really got this. So these differences between protein peptide and peptide spectrometric FDR will vary. How big these differences are will very, very much depend on the size of your data input. So the smaller the number of runs that you combine into a library will be, the smaller the differences. It really becomes a problem in the moment when you work with many shotgun runs. Okay, so it's very important to be aware that these are not set in stone. These numbers that you see there, it will always be like this. Just the opposite. It will depend on the number of input samples.
Absolutely. So this should be evaluated case by case. All right. So where were we? Oh yeah. So if you want to, I mean, all of the details and a bunch of checklists are written in this Nature Protocols paper. So if you're interested in this, I would encourage you to take a look. Okay. So what about assay library aggregation? You know, we realized at some point that we were doing a lot of SWATH projects. So here's SWATH project number 1. We got to do SWATH analysis number 1, and in order to get from this to this, we need assay library number 1, right? So this is where this DDA analysis is happening. And then we did number 2, and then we did number 3, and at one point we said, oh, well, maybe we should try to aggregate these assay libraries together and try to reduce the workload and, you know, potentially disseminate these out to the community. And so this is what we've started to do, and the main one that we've been has been kind of finalized, well, not finalized, but the main one that we've published and is used a lot is this large-scale human assay library. So essentially what we did to construct this was to take all of the DDA runs that we had for any kind of human sample in the lab. So this was a mixture of like cell lines, also this NCI 60 cell line panel, many different tissues that we had analyzed, blood, whatever, purified, everything that we had basically, and different fractionations. So some affinity purifications, some protein fractionation using size exclusion, some peptide level fractionation using 1D gels or off-gel electrophoresis. And we just took everything and we counted up how many DDA files we had, and it was about 331. And we said, okay, we're going to search these all together and see where we get to, right? So we're going to generate an assay library. So we just basically went through exactly the procedure that I described with all this data. And here's the results. So we could identify, using also these strict kind of Mayu-based protein false discovery rate type of approach, we could identify around 10,000 proteins. This had about 140,000 proteotypic peptides and almost 200,000 precursors. And when we turn this into an assay library, if we take 6 fragments per peptide, then you have about 1.2 million transitions in this library. And this is like the 10,000 proteins. This is how it sits on the SwissProt list of proteins that have protein level evidence, and the whole SwissProt. So it's about a half of SwissProt and 2/3 of the ones that have protein level evidence. And essentially, what we wanted to ask as part of this paper was, can this library be used at another lab, right? So at the time that this was done, there wasn't that much SWATH data around at the time from other labs, but one SWATH data set of SWATH data that was available was from Ankel Jangra's lab. She did affinity purification studies, and we decided, and she'd already published this paper, right? So we said, okay, we're going to take our library, we're going to take her SWATH data, and we're going to see if we can reproduce her results. And we just took out the 150 proteins of interest that were used in this study, so we pulled these out of our library. We redid the targeted analysis on her data, and essentially these are the results. So the pink and the green are the data from her library in 2 different conditions. So the details doesn't matter, it was a comparison of some mutants, but these are the protein level fold changes versus a control in 2 different mutants, right? Pink and the green is her results using her library, and the blue and the purple are the same 2 samples using our library. And what you can see is that we essentially, at the protein level, so we extract a different set of peptides, right? We don't have an identical set of peptides in our library as in her library. In fact, she had much better coverage because she did a lot of affinity purifications and she had very high protein sequence coverage. But the point is, we kind of tried to make is that the conclusions at the protein level were effectively the same. So the biological conclusion of the experiment was that these mutants, knock out the binding of this particular protein to a protein complex, and this is what we wrote into this paper. So the basic idea works. And now I'm going to come back to this slide that I already showed yesterday. This is now a larger kind of scale a demonstration of this. So I already mentioned and Rudy mentioned, we had these HEC-293 samples that we shipped all around the world. They acquired the SWATH data at their own sites. They sent it to us, and we tried to analyze the SWATH data with our assay library, right? This big 10,000 protein human assay library. So these different sites didn't do any assay library generation. We just said we're going to rely on our assay library that we already have in-house. And the kind of message is that we basically think this works. So this is now all 5600 data. The chromatography was kind of fairly similar in this kind of thing, but it seems like it's basically this portability question. There's good indications that people can use these libraries that we create here, or other people create. They could be disseminated. So this is kind of what I told you already yesterday. Okay, so last point, I think, second last point maybe. I think I'm running a little bit over time, but I'll try to speed up. But yesterday I told you that if we do these very large libraries, so we have 140,000 peptides, we can only detect about 30,000 of those. So out of the 10,000 proteins we detect about in the library, we detect about 4,000 proteins when we use this large library to do the data analysis. But I mentioned that it's very important to have so the error propagation is a bit different than it is with smaller libraries. And I want to sort of try to illustrate this. So in this library, pan-human library, or this big library is referred locally as the pan-human library, but in the paper it's called combined human assay library. I don't really know why the difference is, but so I might say both. So what I want to try to point out here, this histogram has been shown to you several times by Ludovic yesterday. So this is the one swath file extracted with two different libraries, right? So in the top, it's sort of a sample-specific library that was generated side by side from this sample. It was a HeLa sample, HeLa cell lysate, and essentially, and the second one is when we use this very large library, right? So I don't remember what the number of peptides in this library is, but it's probably 30,000 or something, and here we have 200,000. And this is also what Ludovic was mentioning to you yesterday. So the white is the targets, the red is the decoys. In this type of analysis, where we've generated the library side by side with the swath analysis, where most of the targets are over here on the right. So this means these assays are all identified. So more or less everything that we have in the library is identified in the swath analysis. This is very different to the situation where we use this very large scale library where we have 200,000 peptide ions in the library, but we can only detect about 40,000. So that means you still have this false target. So this white part of the distribution is the false targets, and these are the true targets. So it makes sense, right? The library is much bigger. We don't expect that everything that we had in this huge library that came from many different tissues that we can detect everything. But what I'm trying to get at now is that the point is that this is a very much larger search space in this experiment than it is in this experiment. And now we are going to come back to this propagation of errors from the, well, it's not a PSM anymore, it's a swath assay, but from the swath assay level up to the peptide level, up to the protein level. You have the same problem that we basically described earlier, this error propagation problem. So what I've done now is I've extracted a similar plot as before, the peptide ion FDR, the peptide FDR. So peptide ion means, you know, a peptide at a given charge state, the peptide FDR, the protein FDR. And what you can see is that, and in this case we don't have I profit, but we have M score, which is the score that's coming out of the open swath analysis. And here a low M score is good, right? So as you relax this scoring threshold of the M score, you can see that when we do this sample-specific library analysis where everything in the library we expect to be able to more or less detect it in the swath analysis, we get this error propagation between these different levels is relatively modest. You can almost ignore it, right? So you could almost say at 1% peptide FDR, you know, the protein FDR is only 2 or 3% or something, so this is not such a big deal. When you go to this combined human assay library, now if you say 1% peptide FDR, your protein FDR has gone completely out of control. And this is really, I would really urge people to think about this if they decide they want to go and download these very large scale libraries, and then they plug them into Skyline or they plug them into OpenSwath, and what's computed in Skyline and OpenSwath is an FDR, what Ludovic described, this Q value is an FDR at the peptide ion level. And you say, oh, I'm going to filter this at 1% or 0.01. If you use this very, very large library, then the protein FDR will have gone bananas. This is a very extreme case, but I think it's very important to keep an eye on this. And we're now at the point where we're just figuring out these problems in our lab in the last year, and we've been using now Mayu as well, the same tool that we used in Shotgun to do this error propagation analysis up to the protein level. But it's very much, I would say, kind of an open question, and we're trying to figure out what the best way of doing this. But I just really want to emphasize that if you go for these big libraries, you need to be very careful. And what's important actually is the prevalence. So it may not necessarily be the size of the library, but rather the prevalence. And by prevalence, I mean how many of the targets in your library can you actually detect? And here it's like 20% or something, and here it's 90%. And so this means if we have 40,000 peptides that we can detect, we have 160,000 that we can't detect, and these are all opportunities to randomly get a good hit, and this will add up at the protein level. Okay. This is another way to look at that. So this is the protein false discovery rate plotted as the number of as a function of the number of true positive proteins. The red line is when we use this sample-specific library, and the blue line is when we use this large-scale library. And you can see that the red one saturates, basically. Even as you start to put the FDR out, you don't get any new proteins, and this is kind of reassuring. But this extraction with this large-scale library just kind of keeps going up forever, and this is another way to evaluate this. So this is Tina's slide about Assay library repositories. There are many of them. I'm going to focus on this one called SWATH-Atlas. This is the one that we've been using. So this is maintained by our colleagues at the Moritz Lab at the Institute for Systems Biology in Seattle. And I just want to point out that it's there. This 10,000 protein library is available there. We also have a very comprehensive Mycobacterium tuberculosis library and this is made from analyzing real bacterial lysates and fractions and also synthetic peptides, and it's very, very, very high coverage. There's also a yeast synthetic peptide library there, but more things will be coming here soon, hopefully. As we produce these, then we will kind of release them, use the SWATH-Atlas portal to release them. So the assays come in this CSV format or TRAML format compatible with Skyline or PeakView or OpenSWATH or whatever. Spectra libraries are also available, so one step back in the Spectra format, and you can download subsets as well. You can enter a list of protein accessions and just take the libraries for whatever your set of proteins of interest is. I think I'm way over time. Should I stop, or I can finish this in like 5 minutes? Yeah. Okay, just quickly to make a few comments about moving between different instrument types. So this question has come up a few times, and I just wanted to mention it. It's not a solved problem at all. So there's a couple of papers back in the day saying you can use, in the context of SRM, you can use Y-type ions generated in an ion trap, and they will more or less work on a triple quad. I don't know if people really still completely believe this or not. Another paper saying you can use Orbitrap HCD spectra to go to a triple quad. This is probably more reasonable. And also, Agilent and Waters pushed very much were pushing that you could go from QTOF to triple quad, especially if it's the same vendor instrument. And this basically works. And this again is, everyone asks, can we use the libraries generated on the triple TOF to analyze data on the QExactive? And we don't really completely know the answer to this, but what we do have is this tool, the spectral similarity tool, so we can make a few comparisons and at least make a few comments from it. So if you take the spectra from an Orbitrap Elite in HCD mode and you compare it to a triple TOF in SWATH-MS mode, then this is the agreement. It's actually pretty close to the triple TOF versus the triple TOF, right? So this is the closest, triple TOF DDA versus triple TOF SWATH. And here the agreement is very good. Orbitrap HCD is also pretty good. Orbitrap in CID mode, it gets much worse, and this is also an Orbitrap XL in CID mode. So what I want to point out, this is not really a black and white situation. It's not that you can or cannot use these libraries in one place or another, but there's kind of a continuum of how well they agree with each other. So what we would say is that, like, ideally the libraries should be generated on the same instrument, but that doesn't mean that libraries generated on the next instrument with a HID collision are entirely useless. So you can, basically what I'm saying is you can try it, and the way that the mProfit scoring system is set up, that it's only one of the scores, right? So other scores may compensate for this. So some instruments are more similar than others. HCD seems to be kind of quite similar to QTOF. This is more or less expected. If you want to make these comparisons yourself, you can use this tool, but really the impact on scoring and peptide detection is not really fully evaluated. Very quick comment about IRTs. So this is some data from this cross-lab study where we we analyzed this standard sample and we used slightly different chromatographic setups. So we used 30 centimeter packed emitter tips. Some people were using this Exogen chip LC, but they're all 30 centimeter columns with kind of 3 micron beads. So a little bit different, but not that different from each other. And what I'm plotting here is the errors, the delta retention time. So in the SWATH data, where the peak showed up, how far away was it from the expected time? And you can see you have some distribution where the standard deviation is 100 seconds here. And this is our data, so we expect it to be reasonably tight because we made the library here. If you go to a different site, then this distribution spreads out a bit. If you go to a different site, it spreads out a bit more again. And this is data collected over a whole week, so it seems like they had, probably at the start of the week they had one bias, and at the end of the week they had another bias. But I guess what I'm trying to point out here as well, that it's again not a black and white thing. It's more continuous. It's not that you can or can't use these libraries in a portable way from one site to the other. It's just that maybe the error is a bit bigger, and you need to kind of take this into account when you're setting up the extraction windows and stuff for the data analysis. So in my view, same as this really. These Spectra libraries are portable, but they might not agree quite as well, but that doesn't mean that they're not useful, if you know what I mean. Okay, so I will wrap up here, finally. It took a bit longer than I thought. So assay libraries are essential for SWATH DIA data, is what we would argue, unless you're going for one of these untargeted methods that we discussed yesterday, but these are pretty new. All the details are in this Nature Protocol paper. Try to control the FDR in the assay library generation so you can avoid propagating errors from DDA to SWATH or DIA. The assay libraries are portable if you have the same similar instrument setup, and they may also be portable further than that. Using large libraries can lead to protein FDR inflation, so be careful if you use these large libraries where you have low prevalence that you also compute these false discovery rates at the protein level. I mean, this isn't really implemented directly, for example, in Skyline, but it will come at one point because not many people are doing this workflow, but just want to warn you that you need to think about this if you're going to go for it. And this kind of assay portability is not so straightforward. It seems like instruments with beam type CID are fairly similar, but there are tools available for kind of making these comparisons yourself. And so this is kind of somewhat still open question, I would say. All right, sorry for going over time, but I will try. Maybe we can, are there any questions, or should we just get coffee?
If there's an urgent question, we can do it now. If not, no urgent questions?